data. So biological pathway here indicates an older series of molecular events that leads to a new product or change in the cellular state. And there are many pathway databases in public that classifies those pathways into their function, providing a diagram to see that which genes are involved in a certain process or how they interact with each other. So the important point is pathway is composed of multiple genes interacting with each other. And usually pathway analysis use those pathway information such as gene list or gene-gene relationship from those public databases, and also gene expression data from users to generate the pathway analysis result. So the pathway analysis usually performs a statistical test on each pathway to score and rank the pathways to identify dysregulated ones by stimuli or disease conditions triggered by the experiment. And the pathway analysis started with the gene-based approach, such as GSEA. Those type of analysis used gene-level statistics from individual genes, such as p-values. And around 2009, there was a method called SPIA, which used gene-gene relationship from the databases, and those type of approaches construct a graph with a nose and expressions mapped into that nose. And recent pathway analysis tools are narrowing down the focus into subpathways. So a subpathway means a part of the pathway that's regulated by the stimuli or disease conditions. So until then, there was no pathway tool for time series transcriptome data. So how can we detect subpathways from time series transcriptome data? If it is general transcriptome data, it can be done by mapping DEGs, differentially expressed genes in the pathway to see by comparing control and treatment samples. But in time series transcriptome data, it is hard to define DEGs because it can be detected at each time point or considering whole time points. And as you can see in this diagram, you know the direction of edges. So you have to find the agreement between this pathway information and the actual expression propagation along time. And the second issue is if you are to find regulators of the genes, usually there are a lot of methods to find relationship between regulators such as TFs and target gene by their expression pattern, such as correlation. But in the time series transcriptome data, there could be a delay between the two expression patterns of TFs and genes, and there would be a lot of transcription factors predicted by the method because there are a large number of genes, pathway genes in the pathway, in the significant pathway. So you need to score and rank those TFs to see the important regulators. So these are my two main ideas for these two questions. And first, to detect the subpathways, we use cross-correlation between two differential expression vectors. And to find the regulators, we introduce the influence maximization technique in the time-bounded network. And this is the overview of my work, and I will briefly introduce each step in the following slides. And the first objective is to find subpathways from time series transcriptome data. So before that, we assume that we have a pathway network represented as a directed graph with nodes and edges from CAC pathway database, and we assign a vector for each node, it represents the differential expression as 1 or -1 or 0. So in this example, you can see that for a data set with 2 conditions and 5 time points, you can calculate the differential expression vector with length 5 by comparing 2 conditions at each time point. And this statistical test is usually done by Lima or DSeq software. And so you know the pathway topology, and you have the expression vectors in each node. So to find the subpathway, you need to eliminate some invalid edges from this pathway graph. So to do so, we use the cross correlation here, and the validity of edges can be calculated by cross correlation, and it can see the direction of propagation and the number of delayed time points between 2 genes. So in this example A here, in the valid edges, there are 2 genes with 2 expression patterns, which is very similar but with a positive delay. And in invalid edges, there are expression patterns with negative delay, so we can get rid of these edges from the original pathway graph. And after we find the subpathways, we are to find regulators of those subpathways. And to do so, to search for upstream regulators of the subpathway, we need a bigger network, including TAPs, including a path from TAPs to subpathway genes. So here we constructed an integrated network of gene regulatory network and protein-protein interaction network. And here we used 2 public databases, HDR-IDB and string database. And using the same methods in the previous slides, invalid edges are filtered by the cross-correlation to make it as a time-bounded network. And in the diagram, green nodes indicates genes in the perturbed subpathway. So subpathway genes are mapped into the network to label the starting point of the perturbation. And the final objective is to find the most influential TFs on this network. Here it is colored in orange. And to do so, we integrate the influence maximization technique here, which has been used for social network to find the marketing targets. But the problem here, it is changing to the finding most influential TFs on subpathway genes. So by label influence maximization technique, we can score and rank the TFs by the amount of influence. And as a result of time TP construct a diagram like this, which is called a TAP pathway map and time clock. And it shows a path from TAPs to subpathway genes. And you can see those nodes. You can see the differential expression pattern in the nodes in clockwise direction, colored with blue and red, which means underexpression and overexpression at each time point. And you can see the expression is propagated through the edge to the subpathway gene. And we tested time TP on 2 experimental data. The first was RNA-seq data set of MCF10A cell line with wild type and PIK3CA knock-in samples, and it is designed to activate the PIP3 signaling. And the second data set was microarray data set of EWS-FLI1 knockdown, which is an oncoprotein of Ewing sarcoma, and the paper was to show a co-regulation of this fusion gene and E2F3. And as a result of the first data set, we found that the perturbation in PI3K, AKT signaling, and the consequent changes in downstream pathways of that pathway. And the major findings include FOXO4, which is the known target of AKT, and FOXO and Wnt signaling pathway affected by those FOXOs. And you can see there was a feedback loop in the AKT signaling, which is the reproduction of the same result from the original paper and of the data set. And among those TABs that predicted by time TP, there were 3 important TABs that mentioned in the original paper. And in the second data set, we found out some signaling pathways and adhesion-related pathways, which is the known target of that fusion gene. And because we do not have that fusion gene in the GRN database, TimeTP instead found out the FLI1 gene as a main transcription factor for this data set. And in the downstream of this network, there were 2 important genes, GLI1 and FOXL1, which is the known target of the fusion gene in Ewing tumors. And when we added E2F3 gene in the network, it is predicted to target the same gene with FLI1 or ETS2, which indicates a co-regulation between E2F3 and EWS FLI1 gene, which is the reproduction of the same result from the data set. So in summary, TimeTP uses cross-correlation for estimating expression delay between 2 genes to find subpathways, and it uses influence maximization technique to find and rank the TABs for subpathway genes, and it visualizes the pathway perturbation dynamics for providing a TAB pathway map in time clock. And we provide our software in the website and GitHub. And this is our lab member, and there are 3 fundings and also High TC that provided the travel funding to ISMB. Thank you.